Hello everyone, my name is Gabriel Luiz from USP, and today I'm going to present our study, Pedigree-based evaluation of genetic diversity in Megathyrsus maximus. The Megathyrsus maximus is also known as Guinea grass and is a perennial forest grass widely used in beef livestock systems, especially in tropical and subtropical regions, like Brazil. This species is it's a species of great economic importance, but breeding progress has been limited by the complexity of its polyploid genome and the inherence of some agronomic traits, and because these genotypes occur in both sexual and apomictic forms. However, we didn't have a clear view of how genetic diverse this population really is and how related these genotypes are. So, our main goal was to analyze the pedigree structure of this population to understand the genetic relationship among individuals and also to identify potential inbreeding or imbalance in the genome contribution across the breeding base. We evaluated a breeding population of 960 individuals, include both sexual and apomictic genotypes. Pedigree information was processed using the R programming language with the package AG8 matrix and ASR genomics. To estimate co-ancestry coefficients and also inbreeding and to perform a PCA, we used the Kerr methodology to build an adjective relationship matrix, the A matrix, for polyploid species. This approach allowed us to explore how information flows through the pedigree and help us to detect which parents are contributing the most of the population in the genetic base. From these 960 individuals, we obtained informative pedigree data just from 638 genotypes, including all those that were used as parents, even they don't have information from male or female parents. We found that only 19 genotypes were used as pollen donors and 20 as pollen receptors across all the recorded crosses. However, the contribution is highly unbalanced. The most frequent genotype used as pollen donor accounted for 23% of all crosses, while the second one accounted for 10.5%. Similarly, the two most common pollen receptors were represented by 17.5% and the second one were represented in 14.4% of all crosses. We created this matrix considering a 10% double reduction, and we also detected three genotypes with inbreeding value inbreed coefficient of 0.367, resulting from a cross between a sibling and its parents. Other genotypes showing moderate inbreeding values around 0.1 and 0.12. In the PCA, the first two components explain 16.4% of all the variability of this population, and the first five components together captured almost 30% of all the variability. This pattern suggests that the genetic variability is concentrated with specific subgroups. Our finds reveal that a few genotypes dominate the genetic base of Megathyrsus maximus in this breeding program. This unbalanced contribution can lead to a reduction in the effective population size and increase the inbreeding, especially when we have double reduction. By identifying these issues, we can help the breeders to restructure mating plans and also to promote great diversity in a long-term genetic program and also to make possible to have gains in this process. The take-home message of all this work is to understand The pedigree structure is the key to optimize breeding decisions. I would like to thank CAPS for the financial support and also to Embrapa Beef Cattle team for providing all the data and all the support. Thank you for your attention.